I'd like to thank Dr. Osama, Dr. Khaled for giving me the opportunity to present a quick overview about non-viral gene delivery system. My name is Kareem Ebeid. I'm an assistant professor of pharmaceutics. I received my PhD from University of Iowa in 2018. Here in Egypt, the title is lecturer of pharmaceutics, which is still the same. So I'll be talking about non-viral gene delivery systems. So just to give you an overview about what we are going to talk about. So the contents of my talk, I'll be talking about what is gene therapy. Then we will go to different types of gene delivery systems. As I'm going through my presentation, I will also present some FDA approved gene therapies. Then I'll be talking about the barriers against the delivery of non-viral vectors. I'll give a specific talk on some of the lipid-based non-viral vectors. Then I will end my talk by talking about the optimum properties or characteristics of non-viral gene delivery systems. So let's take it step by step. So what is a gene therapy? If you look at the FDA website, you will see the picture on the left here. So in a gene therapy, what are you trying to treat here? You have a cell, and this cell has a defective gene. And as a cell have a defective gene, it eventually have an error, it have a disease. So what we are trying to do is we are trying to introduce a healthy gene, so these cells can restore its function and you can treat a disease. If you look at the FDA website, you will see the picture on the right. Here, you have a healthy gene that you want to introduce to a patient. So you take the gene, you use a viral vector, and this is what's common, what's present in the clinic right now. You insert this gene in the viral vector. So you have a viral vector, which is a virus, a modified virus that carry a specific gene, the healthy gene, and then you try to insert this viral vector in a patient that have a defective gene in order to restore its function. So this is what they mean by gene therapy. So if you look at the FDA definition for gene therapy, and FDA is the Food and Drug Administration in the United States of America. So when you look at the gene therapy, it's a technique that modifies a person's genes to treat or cure a disease. So you have a patient that have a defective gene, and then you try to modify this in order to treat a disease. And gene therapy can work by different mechanisms of action. So for example, you have a disease-causing gene. So you have a gene that causes a specific disease. And what you do by a gene therapy is you try to replace this copy of the gene by a healthy gene. Or you try to inactivate or silence a disease-causing gene. And this is very common in cancer, where you have an oncogene, which means you have a gene that get overactivated in a way that it controls or it forces the cells to divide in an uncontrollable manner. So what we try to do is we try to intervene and then we try to inactivate this gene, and it's still called a gene therapy. And the third way of using gene therapy is to try to introduce a new or a modified gene that can help to treat a disease. So basically, you are even introducing a new gene or you are silencing a disease-causing gene. And this is what we are trying to do. So as we are talking about gene therapy, so it's important to understand the concept of a gene from pharmaceutics. So when we talk about genes that we want to deliver can I ask all of you to mute? Okay. So when we talk about genetic materials that we want to deliver from a pharmaceutics perspective, we talk about three different concepts or three different categories. We talk about delivering a DNA or a plasmid DNA. As you can see here, it's usually a circular double-stranded DNA. And this DNA, in order to work, it has to enter the nucleus of a healthy cells, so it can get transcribed to a messenger RNA that get translated to a protein in order for it to function. And usually in our work, when you talk about a DNA or a plasmid DNA, you usually encode this DNA with a protein. And encode means you ask this DNA or you design this DNA to carry a specific gene that results in a protein. So when we transfix this, when you add this DNA to a cell, it results in a production of a protein. And usually in our pharmaceutics work in order to see if it's successful or not. It usually carry a gene that fluoresces, it can give light, it can give the signals that you can see. So when you use a plasmid DNA and you try to deliver it, you usually check the cell if it's fluorescent or not. The second genetic material is a messenger RNA. It's a single-stranded RNA molecule, which once reaches the cytoplasm, in this case, you don't want this, it doesn't have to reach the nucleus, it just works in the cytoplasm, it gets translated to a protein, and the same, it gives you like a fluorescent protein from our perspective. And then the final ones that you can see here is a silencing RNA or miRNA, microRNA. These silencing RNA are a double-stranded RNA molecule that is designed for silencing a specific gene. So when you use an siRNA, and this is most commonly used in cancer cells, is when you have an oncogene and you want to silence it. So you use a silencing RNA that once it reaches the cytoplasm, it binds to this gene's messenger RNA and induce its breakdown. So now we have 3 different types of genetic materials that we want to deliver. And I want to stress on when you use a plasmid DNA in our next slides, we are focusing on having a signal. You will see a fluorescence. When you use an siRNA, usually you use cells that is fluoresce, and then you try to turn it off. And these are both indications that your delivery system actually works. So what are the current gene delivery systems? If you look at the current gene delivery systems, basically they are viral or non-viral. And when I talk about a delivery system, it's a carrier that carry your gene and insert it inside the cell. So this carrier is even a viral carrier, which means that you get a virus and then you insert your gene of interest inside the virus, and then you try to get this virus to transfect your cells. And the word transfect means you get the genetic material across the cell membrane inside the cell. So this is what it means by transfection. The other type of gene delivery system that we are focusing on is the non-viral systems. And these non-viral systems means you use polymers, you use lipids, or use a modified version of the gene, and then you try to get this one to cross the cell membrane. So there are 2 different types. Let's start by talking about the viral vectors. So here I'm trying to show you the FDA approved gene therapies. So what we are going to talk about, it's something that's actually practical. It's something that's actually present in the clinic. So I will give you a quick overview about what gene therapies are currently approved. So in 2015, which is around 5-6 years ago, you get the first approved FDA therapeutics based on a gene therapy. And it's called Emlygic. And this Emlygic is kind of a viral vector, and this viral vector is used to treat cutaneous melanoma, which is skin cancer, where you inject the virus directly to the skin where you have melanoma, and then this results in a destruction of the melanoma or of the cancer cells, and also result in a secretion of what's a cytokine called GM-CSF, which stimulates the immune cells to come and destroy the rest of the cancer cells. And this costs the therapy to use like a therapy for a patient, it costs around $65,000. 2 years later, Novartis came out with a viral gene therapy called Kymriah. And this viral vector is based on, or it's used to treat lymphoma. And these are B-cell lymphomas, where B cells in your body start dividing uncontrollably and start resulting in a disease or cancer. And what this Kymriah do is they take the T cells from the patient, and then they transfect these T cells with a specific viral vector. So these cells, once reintroduced back inside the body, can identify these uncontrollably divided B cells and destroy them. And this costs $470,000 in its treatment. In the same year, another product came to market called Luxturna, and Luxturna used to treat vision loss. So you can anticipate someone who lost his vision because of a genetic disorder. You get a viral vector, you inject it next to the eye or like behind the eye, subretinal injection, and then you can retrieve your vision again. So, and this costs $850,000 for both eyes. In 2017, also Gilead, which is one of the big pharmaceutical companies, came out with a gene therapy called the Yescarta, which works exactly like Kymriah, but for a different indication, for lymphoma. And finally, what was really interesting was a gene therapy called Zolgensma. It came by Novartis. And this one used to treat what's called spinal muscle atrophy, which means that small kids or like these child, they can't control their motor actions, they can't move, or even they can't breathe, and this results in death of these infants. And they have found that if you get a viral vector and you load this viral vector with a gene of interest, because these spinal muscle atrophies are a result because of a deficiency of a specific gene. So if you are able to introduce this gene intravenously, you can actually treat the disease. And the course of treatment is around $2 million. So I'm showing you the prices here because I want to show you that using viral vectors are pretty expensive. However, they are yet available in the market. So we are not talking about something that's not translatable. We are talking about something that can reach the clinic, already reach the clinic, but have some limitations. And this is a full indication of what I just talked about. So what are the current limitations of the viral vectors? The first one is what's called immunogenicity. And immunogenicity is once you introduce a virus inside your body, what happens is it elicits immune response. The immune cells detect a foreign body entering the system and then start eliciting a very strong immune response. What happens is when they first start using a viral vector, by injecting adenovirus in a patient in 1999, the patient died within 2 hours just because of the stimulation of the immune response. So most of the viral vectors are immunogenic, even though currently they were trying to modify and reduce their immunogenicity, but still they have some degree of immunogenicity. The second issue in using a viral vector is their limited DNA packing capacity, which means that the virus have a specific dimensions. You cannot load the virus with more than what it can take, right? So the maximum packing capacity in a virus is around 4.7 kilo base pairs. So you have a DNA, that maximum size of DNA is around 5 kilo base pairs. Sometimes you want to insert genes that are 8 or 9 kilo base pairs in size, and this is not available when you use a viral vector. So you have to look for something else. The third major issue with a viral vector is the difficulties associated with its manufacturing, with how to insert a gene in the virus, and eventually the high cost, as I just showed you, that's associated with its use. So that's why we are converting and we are focusing from a pharmaceutics perspective on developing a non-viral vectors. So these non-viral vectors are not immunogenic. There is no limit for packing a DNA. You can put as much DNA as you want. And definitely they are not as expensive as when you use a viral vector. However, everything comes out with a downside. If it has all of these advantages, so what is the disadvantage of using a non-viral vector? The major problem with using a non-viral vector is its low transfixion efficiency. And I mean by transfixion efficiency is how good this vector is able to get your DNA across, or the messenger RNA or the genetic material across the cell membrane so it can enter the cell and then start working. So these non-viral vectors definitely have a lower transfixion efficiency once compared to the viral vectors. However, recent studies are trying to manipulate, trying to modify the non-viral vectors so they can work in a level that's equivalent to a virus. So what are the non-viral vectors and how do they work? Give me a second to silence everyone. Okay. Okay, it's easier to me to ask you guys to mute yourself. Okay, I think we don't have an issue anymore. Okay. So what are these non-viral vectors and how do they actually work? Okay, so the non-viral vectors can come in 3 different categories. And what I mean by non-viral vectors, I'm talking about the delivery system that will be packed with the genetic material and then inserted inside the body to treat a patient. So the carrier systems that we are talking about is made of lipid, or a polymer, or it can come in the form of a DNA conjugate. So the non-viral vector, the first non-viral vectors that we'll be talking about is lipid non-viral vectors. And this is an example of different lipids that are currently used to form liposomes. And these liposomes, they are nanoscale materials, and a nano, when I talk about a nanometer is 10 to the minus 9 of a meter. And to get this in a context, if you get a hair from your body and you look at the diameter of the hair, if you cut this into 90,000 small pieces, this is what a nanometer means. So it's 1/90,000 of a width of a single hair. And when you look at these liposomes, so these liposomes are prepared in a nanoscale, and these liposomes are spherical vesicles made of a double layer of phospholipids. So you can prepare a sphere, so just to get this and to understand what I'm talking about, you can prepare a particle. This particle is made of lipids, of phospholipids, and then you can load the DNA inside it, and then you can deliver it to a patient, and it's able to transfect the cells. So these are the first vectors that we use to do a non-viral delivery system. The second non-viral vectors are polymer-based vectors. And these polymers, they can come in different size, different shapes. But what is common between all of them is they have tons of amine groups, as you can see here. And these amine groups help in getting these polymers to complex with a DNA or an RNA or genetic materials that have a phosphate backbone. And usually the amine groups of our polymers are positively charged, and those from the DNA or genetic material are negatively charged. And this electrostatic interaction helps in getting our polymers to complex and bind the DNA. The most common polymers in non-viral system, the polymer called PEI or polyethylene imine, as you can see here, this is a branched variant of the linear one, where you can have primary, secondary, and tertiary amine groups. And there is a dendrimer called PAMAM or polyamidoamine, and dendrimer means it looks like a branch of a tree, where again you have different types of amine groups, but you can also have a chitosan, or you can have a cyclodextrin that's bound to a polycation. Polycation means a sequence of positively charged molecules attached to it, again, that eventually can bind and can condense the DNA material and help in its delivery. The third form of vectors called DNA conjugates. And what they do in this DNA conjugates is they get the genetic material and they bound or they get chemical conjugate targeting moiety to these genetic material. And let me put this into context. So what you are seeing here is called GalNAc siRNA, and it's used to treat liver cancer. So liver cells are known to express proteins on its surface, receptors called shallow glycoprotein receptors. And these receptors are specified to detect galactose moieties. So if you want something to enter, if you want a therapy to enter the liver cells, all what you need to do is to add some galactose moieties on its surface. And these galactose moiety will direct all of your siRNA to liver cells. And this is what they mean by DNA conjugates, means we are conjugating a targeting ligand, which is galactose in our case, to our DNA. So once injected in the circulation, it will home your formulation, it will home your system. Home means it will take it, and then it will direct it to reach the liver. And that's what we mean by DNA conjugates. However, even though it shows some success, but you have your siRNA, you have your DNA molecule without any kind of protection, and this results usually in a short half-life of these formulations. So as we're talking about these non-viral vectors, there is specific understanding that I have to declare here is each of these vectors, they don't just work miraculously by themselves, but they have to overcome a set of barriers in order for them to function properly. It's not a matter of just mixing anything with a plasmid DNA and injecting it in the circulation so it will work. No, there is a set of barriers that you have to overcome. So what are these barriers? So our next part of our presentation, we will be talking about the barriers that are currently available in your circulation, in your body, that prevent any of the formulations from working actively. And then from our, as a formulation scientist, we are trying to overcome these barriers in order to succeed in delivering our gene. So if you look here, you will see this is a picture depicting a blood vessel. This is a kidney. And it's called here extracellular barriers, which mean the barriers outside the cells. So we are dividing these barriers to extracellular barriers and intracellular barriers that we want to overcome. So if you look at the pictures up here, these are kind of delivery systems that we are preparing. These are nanoparticles, these are liposomes, or these are whatever kind of delivery system that we are trying to get to reach their target cells. So what barriers do we face when we are trying to deliver our formulation? The first one is called endonucleases. And endonucleases, these are enzymes in the circulation that break any kind of foreign genetic material. So if you are delivering your formulation inside the body, you have to make sure that your gene is fully protected, your DNA or sRNA is fully protected from these endonucleases. So you have to keep this in mind when you start designing your delivery system. The next issue that you face is the detection of any foreign material by the immune system. So once you deliver your genetic material, once you deliver here your delivery system, non-viral delivery system, the first encounter in the circulation are the macrophages, which are specific immune cells that detect any foreign material. And if the macrophage is able to detect your delivery system or your non-viral vector, it will capture it, it will engulf, and it will destroy your formulation. So you have to find a way to evade, to overcome the uptake of your formulation by the macrophages. The third issue that you face when you inject a genetic material is the renal excretion. So usually, any material that's below 10-20 nanometer in size, that's very hydrophilic, tend to get excreted from the circulation through the kidney. And most of genetic material, if you just inject a messenger RNA, an siRNA, a plasmid DNA, it will eventually end in the kidneys because they are pretty hydrophilic, they are pretty water-loving molecules, and they are pretty small in size. So you also have to keep this in mind that if you are preparing your formulation, you have to design it in a specific dimensions so it is larger than this 20 nanometer in size, so it's not going to get excreted through the kidney. The fourth major barrier is called extravasation. And extravasation means you get your formulation to move outside from the circulation and then start moving and directing towards the target cells. And usually this extravasation is also size limited. And when we talk about tumor cells or cancer cells, which is our major focus when we use these non-viral vectors, these blood vessels have gaps, have fenestrations of a specific size, that if you meet the size, you will be able to get your formulation or your delivery system to extravasate, to move outside the blood vessel and start moving toward the cancer cells. So when you design your formulation, your delivery system, you have to keep all of these parameters in mind. Are we done? No, because once your formulation reaches the target cells, you get extra sets of barriers. The first set of barrier is a specific uptake. So you need your delivery system to be taken up specifically by your target cells. And this is usually done when you have a ligand to a receptor. So you have cells that have a specific receptors, and then you add a ligand or you add a targeting moiety that can bind specifically to these targets and then taken up by the cells through endocytosis, which is called receptor-mediated endocytosis. And this is exactly what happened when you use a GalNAc in the examples that I just explained. So also you need your delivery system to home to target a specific cell population. You don't want your delivery system just to attack all of the cells in your body. And this is basically done through adding a targeting ligand. The 6 major issues that you face when you design your delivery system is the presence of your viral or your delivery system inside the endosomes. And these endosomes are vesicles inside the cells that have harsh pH conditions where the pH inside these endosomes are pretty low, it reaches almost 2. And usually this pH activates specific enzymes that degrade anything inside endosomes. So if you want your formulation to work actively, you have to make sure that it can escape from the endosome, or it can achieve what's called endosomal escape. Finally, if your formulation is able to escape from the endosome, you have to make sure that it can release its cargo. It can release the DNA or the messenger RNA or the siRNA inside the cells. Because if you have delivery systems that's highly bagged and highly condensed and bound strongly to your DNA or your genetic material, and it reaches the cytoplasm and it's not able to release your genetic material, then it will definitely not work. And finally, so what's called nuclear translocation. And this is specific for the plasmid DNA, because for a plasmid DNA to work, it had to reach the nucleus, it has to cross the nuclear membrane, and then it has to go to the nucleus so it can undergo transcription and translation. However, for a messenger RNA or siRNA, they work in the cytoplasm. So when you use a plasmid DNA, you have to keep in mind that you have to make sure that your plasmid can eventually reach the nucleus. So these are 8 sets of barriers that a formulation scientist, especially in a pharmaceutics department, has to consider when he designs his delivery system. So now how to overcome these barriers. So now I explained the barriers. Now from a pharmaceutics perspective, which is an important part of my talk today, is how can we overcome these barriers step by step. So let's take them once by one. So the first barrier that we talk about is endonucleases, which are these enzymes that once find a different or like a foreign genetic material, they attack and destroy it. So what we usually do here is you have a genetic material that is negatively charged, and this is a DNA, and it's negatively charged because it has a phosphate backbone. And what we do is you use your non-viral vector, your positively charged polymer or lipid, and it's positively charged because of tons of ionizable amine groups. And once you complex them together, once you mix them together, they form an electrostatic complex where you have the negatively charged phosphate molecules attached to the positively charged amines. And this results in a condensation and eventually result in a shape that you can see here on the right, where you get these polyhedral particles. They are pretty small in size, around 40 nanometer in size, and they condense the DNA material inside it, and they protect it from complete degradation. So the first part, in order to overcome endonucleases, all what you need to do is you need to complex your genetic material with a positively charged polymer, and the result is a nanoplex, as we call it, and it's completely protected from endonucleases. And let me show you how to actually show this. How, if we prepare our nanoplexes, is it actually able to protect our DNA from degradation or not? So as you can see here, this is an experiment where they get a double-stranded siRNA molecule, and they added a fluorescent probe in one side, and they added a quencher from the other side. A quencher is a molecule that prevents the fluorescent probe from fluorescing. So if your siRNA is intact, the quencher would inhibit the fluorescence, so you are not going to see any signal. So if your formulation or if your nanoparticles is working and is capable of protecting your siRNA from the nucleases, then you will never see a signal. However, if your siRNA breaks apart, then the quencher and the fluorescent molecule will fall apart from each other, and then you will see a signal. So what I'm trying to show you here is if we see a signal, it means the DNA or the siRNA get damaged and the endonucleases works. And if we didn't see a signal, this means that we are successful in preparing our formulation from endonucleases. And this experiment was done as follows. They get a tube where they add just siRNA by itself, and definitely this will not fluoresce. And then they add nanoparticles by itself, and this will not fluoresce. And then they add an siRNA plus RNases, so which is an endonuclease that breaks the RNA, and they incubate these for 5, for 10, or 15 minutes, or they get our nanoplexes, our formulation that's protecting our siRNA, and incubate it for 4 hours with endonucleases. And these are the results that we achieved. On the y-axis, you have the intensity or have the degree of fluorescence. And then what you can see here is when you have your nanoparticles, which is blue, plus RNAs for 4 hours, you can see this curve down here, which means there is no fluorescence, which means that your nanoparticles were able of protecting your siRNA from degradation for almost 4 hours. However, if you look at this curve, the orange one, which when you have the siRNA plus RNAs for just 15 minutes, You can see a huge increase in the peak here, which is the orange peak, and this indicates the breakdown of the RNA. So the message that I want to deliver here from this figure is when you complex your genetic material with a positively charged delivery system and you condense it, you are capable of protecting it from endonucleases. So it will achieve a longer circulation half-life, and then you will have higher chance of your formulation to reach the target site. The second issue that we face is how to evade, how to overcome, how to ask or how to design your formulation to hide from the immune cells. And this is usually done by adding a polyethylene glycol layer. So you have your nanoparticles here, or nanocomplex or complex system, that have your positively charged polymer siRNA, and then you can chemically modify it by adding a polyethylene glycol layer, which is just a bunch of carbons and hydrogens and oxygens. When you add these polyethylene glycol layers on the surface of your formulation, you are going to increase the hydrophilic properties of your particles, and increasing the hydrophilic properties of your particles usually result in evasion of immune system and longer circulation half-life. And you can see it from here. So they did an experiment where they had a polyethylene imine, the PEI polymer, they radio-labeled it. And then they have a polyethylene imine conjugated to polyethylene glycol. And both of these were complexed with plasmid DNA and injected in a circulation of mice. What they did here is at each time point, on y-axis you have different time points. At each time point, they measured how much is remaining out of the injected dose. And what you can see here is when you have PEG, polyethylene glycol layer, you still see the same concentration of the polyethylene imine over almost 120 minutes, over 2 hours, which is more than enough for your formulation to reach a target tissue. On the other side, if you don't have PEG, you see a quick decline in the concentration of the PEI, which indicate the uptake and removal of PEI from the circulation using the immune system. So the second way of improving your formulation is to add a hydrophilic polymer on the surface of your formulation to increase its circulation half-life and evade or overcome the immune system. The third issue that we talked about is kidney excretion. So if you just inject siRNA by itself, it will result in accumulation in the kidney. If you look at the picture here on the left, it gives you an indication or a correlation between the nanoparticle size and in which organs do they accumulate. And if you look at here, particles less than 5 nanometer in size tend to accumulate in the kidney. Very large particles, more than 150 nanometer in size, tend to accumulate liver, spleen, and the lung. So if you want to design a delivery system that can overcome liver accumulation and overcome kidney accumulation, you have to design it in a specific size range that can evade or can overcome the kidney excretion. And on the other hand, you don't also want it to accumulate in organs like a liver and a spleen. So you have to be in the intermediate size. If you look at the figure here on the right, which is my figure that I did in USA during my PhD, what we have done here is we get a fluorescently labeled siRNA, and then we injected it at the naked siRNA inside the mice. If you look at the figure here, and then after we injected the siRNA, we collected the organs, and then we use a machine called IVIS, in vivo imaging system, that can detect the fluorescence from each specific organ. So after we inject our formulation, we collect the organs, and then we set it under the machine, and then we start collecting the signals. So the first one on the left is an untreated control, which you usually don't expect to see any signal coming out of it. We have a tumor, heart, lung, liver, spleen, and kidneys. When we inject just pure siRNA, just siRNA by itself, you can see that all of the siRNA accumulate in the kidney, and this is based on being a hydrophilic molecule small in size. However, once we prepared our nanoparticle system, which we call here a PM, so this is a nanoparticle that's complexed with siRNA, so this is a nanoplex system. And instead of having all of your siRNA in the kidney, now you get a distribution throughout all of the tissues, and you get some of your formulation actually in the tumor. So making the nanoplex, increasing the size of your formulation, you have a different distribution pattern of your siRNA molecule. So now if you want to target a different organ rather than the kidney, all what you need to do is you need to complex your siRNA in a formulation. You have to increase its size, and then you will have a different distribution pattern. What is really interesting is when you add PEG, which is abbreviated as a G here, so this is a PEGylated nanoparticle. So this is a nanoparticles complexed with siRNA, pretty small in size, around 100 nanometer, but it also has a PEG layer on its surface that increases circulation half-life. When you increase the circulation half-life of your formulation, you even get a higher signal from the tumor, and this indicates a higher circulation half-life. So again, you can overcome kidney excretion by controlling the size of your delivery system. And if you PEGylate your delivery system, you are increasing your circulation half-life even more, and you have higher chance of your formulation to reach your target site. The fourth barrier is called extravasation. How can you get your formulation to cross the endothelial cells, the blood vessels, and get out from the blood vessels to reach the tumor cells? And this is specific for tumor cells and for inflammation. The major issues that you have, or it's not a major, it's an advantage that you see in tumors, is when the tumors grow, they usually grow in a very fast rate, and they send a lot of angiogenic signals. They send signals to the surrounding blood vessels so they can get support, or they can get blood so they can continue their progression and division. So the blood vessels that support the tumor cells, as you can see here, so these orange cells are tumor cells. The endothelial cells that support the tumor cells, they have gaps, and these gaps are somewhere between 50 to 150 nanometer in size. And these gaps are not present next to healthy cells, as demonstrated here by the green cells. So if you get your nanoparticles or nanocomplexes by a specific size, somewhere less than 200 nanometer in size, and you start injecting them intravenously, what happens is your nanoparticles or your gene therapy get bounced throughout the blood vessels, but it's able to extravasate through these fenestrations, these holes in the blood vessels, and they can accumulate in the cancer cell, and eventually will get a higher accumulation in the cancer cells, so you are kind of targeting these cancer cells. So extravasation is achievable once you control the size of your formulation, and it's pretty advantageous when you're targeting cancer or targeting inflammation when there is fenestrations in the blood cell. And again, this is demonstrated here, where in the same figure you get a good amount of tumor accumulation here and here, basically because these tumors have defective blood vessels that have fenestrations. Our formulation here that's used in this formulation, that's used in this experiment, was around 100 nanometer in size. So this 100 nanometer was able to extravasate the circulation, accumulate in the tumor, as you can see here and here. Here is the quantification of the signals that you see. If you look at the amount of formulation in the tumor, the blue is siRNA by itself. When you add it in nanoparticles, you get a higher or boost, an increase in the accumulation in the tumor. When you use a PEGylated formulation, you get a much, much higher accumulation in the tumor because of, again, this increased circulation half-life. So when you have a formulation that's less than 100 or 100 nanometer in size and PEGylated, it will circulate for a longer time. You will have higher chance to extravasate and reach the tumor, and that's why you get a higher signal here. So now we were able to overcome the 4 barriers that represent the extracellular barriers. And then now we have to go to the intracellular barriers. The first one of the intracellular barriers is specific cellular uptake. This group, they design a cationic polymer, as you can see here, and then they complex it with siRNA at NB ratio of 80. And this NB ratio is nothing but the amount of nitrogens in your positively charged polymer to the amount of phosphates in your negatively charged siRNA. So NB 80 means you have 80 more nitrogens in your formulation compared to the phosphates in your siRNA, and this is a way of complexing them. So you complex your formulation with an siRNA, and then you add a targeting ligand called a COPA on the surface of your formulation. And what this targeting ligand do is it force your formulation, your delivery system, your nanoparticles to reach a specific population of cells. And this COPA is prostate specific, means if you have prostate cancer cells, that overexpress a protein called prostate-specific membrane antigen. So this ACOPA will take all of your formulation, it will bind to the specific receptor, and it will direct your particles to be endocytosed only on cancer cells, so you get a higher dose of your particles in cancer cells, and eventually you achieve a higher effect. So how can we test this? So what they have done here is they used a fluorescently labeled siRNA. They have their nanoparticles, which is A, and then they have the targeting ligand, the modified nanoparticles that have a targeting ligand on the surface, the targeted nanoparticles as B, and they did a flow cytometry experiment. And a flow cytometry experiment is that you get your cells of interest, you get the cancer cells, and then you add your formulation on the cells, and then you measure the degree of fluorescence that's coming from your cells. So if your cells are able to take up your fluorescently siRNA, then you will see a shift in the signal, you will see an increase in the fluorescence. If you look at the figure here, the x-axis here represent the level of fluorescence that you are getting from your cells. And as there is a shift toward the right, this histogram represents a cell population. And as there is a shift toward the right, it means that these cells are taking up your particles. So what you can see here is they used a prostate cancer cells that doesn't have or have a low PSMA expression, that the receptor is not expressed to a high level. So when they have the targeted or non-targeted particles, they almost overlay, like the red and the blue histograms almost overlay, which means that there is no advantage of using a targeting ligand in this set of cells, which is pretty interesting because these cells don't have the targeting, or they don't have the receptor on their surface, so you wouldn't expect to see a difference. And the same in the other cell lines. Time? Okay, let me finish this and then we can finish the rest in another time. Let me finish just this figure. And then what is interesting here is once you have a cancer cell that overexpress this specific protein, you will see that the blue curve here shift more toward the right compared to the red curve. And this indicates that there was a specific uptake of our formulation using our targeting ligand. So the sixth barrier is the endosomes. When your formulation reach the endosomes, usually they are exposed to extensive or harsh low pH conditions, which usually destroy your formulation. So you need for your formulation, usually when you use a polymer, they have what's called the proton sponge effect. So they can escape from the endosomes using what's called the proton sponge effect, and I explain this now. And the lipids have what's called the membrane fusions, where the lipids can fuse to the membrane and then they can go outside to the cytoplasm. It's pretty interesting to give you this presentation about the proton sponge effect. So what proton sponge effect means is when you have your formulation here inside the endosomes, these endosomes have their proton pumps, so they start bumping protons inside the cells. And as proton are bumped, also chlorides bumped to neutralize the charge in the endosomes. And then you have a lot of amine groups in your formulation, and these amine groups, what they usually do is they usually take these protons and get protonated. And as the pH doesn't go down, the endosomes start pushing more protons in, more chloride in. The protons are taken up by your formulations. That's why they say it's a proton sponge, because it acts like a sponge to take all the protons in. And as the chloride concentration increases, it's associated with an increase of the osmotic pressure inside the endosomes. Water diffuses in, and then the endosomes rupture, where your formulation can go out and release its DNA content. So that's called by endosomal escape. To see this experimentally, they used the fluorescently labeled siRNA. They used a polymer, with the same groups, use the polymer, make a nanoplex. And then they take the nanoplexes or the formed particles, and then they add it on the surface of the cells. And then they stain different portions or different organelles of the cells, and then they see it, or like they try to see it under the confocal microscope. So here what they did is they used two different polymers. One of them is branched polymer, one of them is a linear polymer. And then they tested both and see which one of them can result in a better endosomal escape. What you can see here is the siRNA is labeled red, the endosome is labeled green, the nucleus is blue, and then you have an overlay. If your formulation is able to escape from the endosome, you would probably won't see any red and green overlapping on each other. But if your formulation are stuck inside the endosome, then the red and the green signal will overlay, and this will result in eventually a yellow color. So for this branched polymer, you barely see a yellow here. However, when you have a linear polymer, most of the signal is yellow. And if we magnify both images, you will see in a branched polymer, you just see tiny yellow spots. However, when you use the linear polymer, you have a big bunch of yellow color here. So this indicates that the branching of the polymer is pretty critical to get a good endosomal escape, and this is expected because you have tons of primary, secondary, and tertiary amines that can capture more protons, and this results in a better proton escape. The seventh barrier is how to release your cargo. Can your formulation, once it reaches the cytoplasm, can it release its DNA content? So how we examine this is when you run what's called the gel electrophoresis. In a gel electrophoresis, you complex your DNA with your nanoparticles or with your polymer to form nanoplexes at different ratios. And what you can see here, and then you turn the gel electrophoresis on, and then if your DNA is not bound to your formulation, it will not migrate. So what you can see here is when you have a plasmid DNA, you see a band down here. When you have your nanoplexes, that complexes your plasmid DNA at different ratios, when you go to the right, you get higher ratio of your nanoparticles, you get higher amount of amine groups, so you can complex your DNA. So at NB5 and 10, you can see a band here, which means that your formulation was able to complex the DNA completely. But can you release it? So what we do here is we add heparin to our formulation at specific concentration. And if our formulation can release our DNA, you will see the band down here. As you can see here, when heparin is added, your formulation is able to release all of its DNA content. And inside the cells, there is a lot of negatively charged proteins like heparin that are endogenously present in the cells. And then once your formulation reaches the cells, they will start binding to your formulation and releasing your DNA content. And finally, the eighth barrier is the nuclear translocation. And this is needed only when you have a plasmid DNA. If you're working with a messenger RNA or siRNA, you don't care about having this. However, when you use a plasmid, you have to make sure that your plasmid can cross the nuclear membrane and enter and reach the nucleus. And this is usually done when you have what's called a nuclear localization sequence. And this is a sequence added to your DNA. And once your plasmid DNA is released, it helps in directing your DNA towards the nucleus. However, the easiest way or the most sufficient mechanism at which the plasmid DNA reaches the nucleus is during the mitosis and the breakdown of the nuclear envelope. Because when the cells start going inside the mitosis, they start breaking their nuclear envelope. And when the nuclear envelope breaks, you can easily have your plasmid inside the nucleus when it's formed again. So there are two pathways at which nuclear translocation is achieved. And by this, I finished my 8th barrier. I still have more to say, but now I finished the barriers.